I'm Dr. Zhang from Tianjin Medical University General Hospital. It's my pleasure to welcome all of you who are interested in neuroimaging research in dementia. Neuroimaging is a crucial method to identify in vivo the structural, functional, and even molecular changes of the human brain in either normal aging or disease. Clinically, routine MRI scan has been used to identify various kinds of dementia, such as Alzheimer's disease, frontotemporal dementia, and vascular dementia, with corresponding criteria based on regional brain atrophy or cerebrovascular lesions. Moreover, although not yet widely used clinically, there are a number of additional informative MRI modalities that can elucidate the pathophysiology in neurodegenerative conditions, such as diffusion tensor imaging, arterial labeling MRI, and resting state functional MRI. In addition to MRI, PET neuroimaging techniques have played an important role in the diagnosis of various kinds of dementia and other kinds of neurodegenerative diseases, with appropriate tracers to assess the pathophysiology in vivo in neurodegenerative conditions. FDG PET is considered to be as a downstream biomarker of neuronal injury and neurodegeneration. Characteristic patterns of regional metabolic changes of the human brain on FDG PET have been identified in the association of common types of neurodegenerative dementia. Furthermore, to date, several PET tracers are available to assess amyloid plaque and tau burden in vivo. These molecular ligands can facilitate to the accurate diagnosis of different kinds of dementia and other kinds of neurodegenerative diseases. Today, in this workshop, we will focus on recent neuroimaging findings, in particular MRI, in persons with dementia, such as Alzheimer's disease, frontotemporal dementia, and subcortical ischemic vascular dementia. We are going to have 3 presentations followed by a Q&A session. Okay, now it's my great honor to introduce our first speaker, Qin Chen. Okay, Dr. Chen is an associate professor of neurology in West China Hospital of Sichuan University. She got her PhD and MD in neurology from West China Medical School of Sichuan University and a research fellow at Center for Advanced Imaging Research of Mayo Clinic in the United States. She is currently a member of FTD Prevention Initiative, a member of the International Society of Magnetic Resonance in Medicine, and a member of the Alzheimer's Association International Society. Her talk's title is Neuroimaging in Familial Frontotemporal Lobar Degeneration. Welcome, Dr. Chen.
Thank you, Professor Zhang. Thanks for the kind introduction. So let me just sharing my screen. So can you see my screen right now?
Yeah.
Oh, that works, right? Okay. Good morning and good afternoon. My name is Qin Chen, a behavioral neurologist from the West China Hospital of Sichuan University. I'm very glad to share the topic of imaging biomarkers in the familial frontotemporal lobar degeneration here today with you guys. Okay, first one, we started with the term of frontotemporal lobar degeneration. This is a pathologic term. It is associated with a heterogeneous group of the clinic syndromes. So we see that here in this chart. We know that including in the clinic, we can see the Ftd spectrum, as well as we can see the FTD-associated syndrome, like motor neuron disease, PSP, and cortical basal syndrome, CBS. So for the FTD spectrum, in many indicated there are three major clinic syndromes, including the behavior variant of frontotemporal dementia, the BV-FTD, as well as the language problems with the PPA, including the non-fluent or agrammatic variant type, or semantic variant type of PPA. But we need to tease out the neurogenic variant of PPA, which we put into the Alzheimer's disease spectrum. So we see the frontal lobar degenerations is a pathologic term.
Sorry?
Okay, I'm sorry.
We cannot see you PowerPoint. Could
You can't see the PowerPoint?
Yeah.
Okay.
Could you share your...
So let me sharing that again. Sorry, I can't see that. I think that works. Okay, here.
Okay, it's working.
Working right now?
Yeah.
Good. Okay, so I think we are on the second slide here. So you see then here we just talk about the pathologic terms of FTLD as a clinic syndrome, including a lot. So we all need to tease out the neurogenic type. So as we'll see, then the pathogenic term of FTLDs were very complex. So even if in the pathologic studies, there are three main pathologic types for FTLD. There are many tau pathology, TDP-43 pathology, and FARS pathology. So even if in the TDP pathology we have different subtypes, and in the tau pathology we have different subtypes, including 3R and 4R isoforms, different isoforms there. So this is very the clinic syndromes we mentioned before, the BV-FTDs, we can have all three kinds of pathology. And as well with the semantic variant PPA, we have the TDP and tau pathology there. And so this makes this disease very complex. So different clinic syndromes mentioned above are associated with different pathologies as shown here in these figures. So it is very complex even for the clinic practice and even for the research. But however, the good news is that there are 30 to 50, many up to 40 of the FTLD patients are familiar. And when I say familiar, I mean that there is a first or second degree relative with the diagnosis of one of the FTD syndromes or a motor neuron disease, ARS, or primary lateral sclerosis, PLS. So the majority of the familial FTLD is accounting for in three main genes: the MAPT gene, the granulin gene mutation, and the repeat expansion of C9 of 72. So that makes this genetic mutation careers, makes the disease more pure and earlier for us to studies here. So this gives us a time window for us to tracking the disease, to describe how the pathophysiology changes in vivo from the asymptomatic to the symptomatic stage. So in the hypothesis, we can see that in the FTLD mutation careers, the biomarkers we mentioned here in the red lines, the green lines here, the biomarkers will change ahead of the symptomatic, the clinic symptomatic onset. So this time windows gives us opportunity to study or treat this disease, even from the normal to the abnormal during these disease progressions. So there's a lot of imaging studies tracking the familiar FTLD families to see what happened of the brain before the symptom onset. So here this is what we meant talk in today's talk. We have mentioned the neuroimaging here. The neuroimaging we applied in the familial FTLD, many including MRI, even if the molecular images we mentioned. The MRIs we divided into structural, functional, and metabolic some subgroups. We'll talk it in the later. So we'll first talk about the structural MRI. So if we know that there's three main gene mutations, right? The first one is the MAPT mutation. The MAPT mutation located in the chromosome 17. There are 6 tau isoforms generated by the alternative mRNA splicing. So we know that within the exon 10, they're involved, when this exon 10 involved, so there are many 4R tau predominant. They are many present with a CBD or PSP. If without the exon 10, there are many the 3R tau pathology, the Pick's disease. So there are actually, with or without the exon 10 involved, so there are at least 3 patterns tau observed in the FTLD tau depending on the mutation sites. So there are maybe the 3R, pure 4R, or mixed both 3R and 4R. That all can be seen in the MAPT mutation carriers. So we know then that different subtypes of MAPT mutation, they were observed this different subtypes of the MAPT mutation. We see then there are different brain atrophy. So this figure shows that structural MRI from the patients who are carriers at MAPT mutation. This one is a V337M, which is the 3R and 4R tau pathology. So this makes some asymptomatic patterns there. There are only, but this patient have 20 years of the disease duration. And this is a 4R tau mutation. The 3R tau mutation, which is very young and only with 1 year disease duration, you can see the atrophy very obvious. And this is a 4R tau. This is IVS 10+3. This mutations. So this patient has 3 years durations of the disease duration. We can see some symmetric atrophies of the hippocampus and the dilation of this ventricle. But this one is another 4R tau predominant with some of 3R tau. So this one is a P301L. This mutation, we can see that these patients only have 6 years of the symptom onset, but the brain atrophy is more predominant than other mutations. So it may not be successful to correlate structural MRI with a biologic aspect of hereditary tau pathology because of the different rates of atrophies and sequences of automatic involvement, which are highly variable even in cases of the same mutation. So that's why we saw their limitations for if we done the studies, you only do the cross-sectional studies. The individuals and different stages of the disease are not comparable. So the initial location and sequences of atrophy are no longer being discovered anymore. So that's why we need to do the longitudinal MRI studies. We can see the rates of atrophy. We can see the different brain regions. We can see where is the initial atrophy begins and how the sequences of the atrophy spread out. So this is what we do. This is a cross-sectional. This is cross-sectional studies that previous has reported. That's what we're doing there. So they compare the asymptomatic versus the normal controls. These are non-carriers, and symptomatic MAPT mutation carriers versus non-carriers. So it has you see that if in the patients in the asymptomatic, there are very nice atrophies in the bilateral hippocampus or anterior temporal lobes. And if they are symptomatic patients, there are more predominant atrophies even in the group comparison. So this is what we did. So this is what in our previous reports, we followed about 14 asymptomatic mutation carriers and 13 non-carriers who underwent structures MRI and were followed annually with a median of 9.2 years. So here we applied this tensor-based morphometry with a symmetric normalized. We saw a TBMC. This is it's a kind of calculated algorithm to calculate the brain atrophy rates, even if the lobar atrophy rates, we can then just include the mixed model to determine the trajectory of the lobar atrophy. So we see here the blue lines is the asymptomatic MAPT mutation carriers. The red lines is the symptomatic mutation carriers. Well, who are converted from the asymptomatic. So the black lines are the non-carriers. So in the top panels, the observed trajectory were plotted for the repeated measures for one individuals in the raw data. Each line is one individuals. So in the bottom panels, the lines show then the predicted volume from the mixed models. Each shows what we could expect per group in the age range. So the x-axis is shows the age. We can see there is an accelerated decline then in the temporal lobes, even if in the blue lines in the asymptomatic mapped mutation carriers. And they are spread more for the mapped mutation converters. They are followed by the frontal and parietal lobes, which are become when the asymptomatic mutation carriers become more symptomatic. This is more steeper. So according to the cross-sectional studies of the mapped mutation carriers, we can see that the patterns is here. Then the mapped mutation carriers showed a brain atrophy patterns is relatively symmetric atrophies, including the anterior temporal lobes, accompanied with laser atrophies of the orbital, frontal, and lateral prefrontal cortex. Okay, the majority of the cases are really, I mentioned, is a symmetric patterns. So this is the characteristics of mapped mutation brain atrophy pattern. So let's go move on to the granulin mutations. The granulin mutations also the most of these FTD patients are familiar with this FTLD, the major causes. These mutations are first identified in the 2006 and result in the loss of progranulin levels through the haploinsufficiency, which leads to an intraneuronal aggregation of multiple disease related to proteins such as TDP-43. So there also can be a lot of clinical manifestations. The BVFTD also can be presented with PPA, also can be presented with the CBS. So for the granulin mutation carriers, we see the cortical atrophy here is more asymptomatic patterns, with one hemisphere is more predominant. So this is as a structure of but in the asymptomatic granulin mutation carriers, previous studies are inconsistent. Some of them find some atrophies in some regions, but others didn't. So especially in the asymptomatic granulin mutation carriers, the gray matter volumes different is not coexistent with each others. So that's why when we have the questions about how the progression of atrophies in the asymptomatic granulin mutation carriers. We need some longitudinal studies here. So this is what we did. So we involved the asymptomatic granulin mutation carriers and do the longitudinal studies to see the brain atrophies there. And here we see this is similar to our previous studies of the mapped mutation carrier. So we show that there's a trajectory of change in the lobar cortical volumes in the black lines and for the non-carriers, blue for the asymptomatic and red for the symptomatic. So the hemisphere volumes were averaged because of average, there is no difference between the left and right lobar cortical volumes in the progranulin carriers in current study. So we see then the frontal and the parietal lobe, the frontal and parietal lobe cortical volumes decline started in the granulin mutation carriers as early as the asymptomatic stage and become more atrophic as the disease progressed in the symptomatic stage. This annual change for the composite volume, so we combine the frontal and parietal lobar volume as one values. We have seen that this is more robust change in the asymptomatic stage. So this case, we see them here. So we use the frontal, temporal, and parietal volumes as the y-axis, and this x-axis we show that the 0 indicates the symptom onset. That we see the 5 years before the symptom onset. We can visually detect there is a decline of the brain volume there. Okay, so let's move to the C9. The repeat expansion of the C9 is also the most common gene associated with the FTLD, especially with those FTLD and ALS kindred, and also the TDP43 based pathology. The C9 mutation career, the C9 repeat expansion careers were thought to involve autophagy and endosome transport. So, different from the granulin mapped mutation career, C9 shows a lot of widespread of the brain atrophies, even if in the asymptomatic stage. It seems that everywhere, and even including the cerebellum. This is different from the other two mutations here. So here is a brain atrophy, but how this brain atrophy pattern is different from each common mutation careers. I think this is a very good studies by Dr. Cash in 2018. And we see that there are 3 mutations types here, 3 genes here. And we see the first line for each gene, the first line is asymptomatic, the second line is asymptomatic. So we see that there are some different atrophy patterns in the symptomatic stage, but more of them are included, the temporal and parietal lobes. But for the asymptomatic, it's a little different. The mapped mutation, we see this, the symmetric, the hippocampus, the anterior temporal lobes, but the C9 with the widespread atrophies, right? Even if the asymptomatic stage. But for granulin, we'll see that maybe asymptomatic here, some regions there, parietal lobes. So we see that structural MRI demonstrated the cortical degenerations with a mutation-specific neurodegeneration pattern years before onset of clinic symptoms in the asymptomatic familial FTLD mutation careers. Okay, let's move on to the diffusion tensor imaging. This is what we did for the mapped mutations, to use the DTIs to do the cross-sectional and longitudinal for the white matter degenerations. We do the mapped mutation we see here. You can see that when we do the whole brain analysis, we see this, this is in line with what we found in the cortical atrophies. We see that the endocyno white matter degenerations here, even in the asymptomatic mapped mutations, increased MD. Then when the symptomatic mapped mutations are wide range, just widespread, even if the whole brain, anterior, temporal, parietal, and frontal lobes. But the FA shows a similar patterns with the MD, but FA didn't show any change in the asymptomatic group comparison. This is a cross-sectional. So we also use this endocyno white matters to predict how we near to the symptom, how we correlated with this MD or FA with this prediction of the symptom onset. So we see that the decreased FA and increased MD of endocyno white matter are associated with the proximity to the symptom onset in the mapped mutation careers. And the trajectory of MD in the endocyno white matter increased faster with agent in the careers compared with non-careers. So for the granulin mutation white matters, they also have seen that is the asymptomatic there, and what diffusion tensor images can detect the white matter abnormalities 2 years before the symptom onset. So this is for the individual cases of granulin mutations. We see that in the clinic cases, we saw that the white matter degenerations here, and we can also see this. There's white matter lesions in the pathologic. There are cortical neurolosis secondary to that. The axon degeneration and demyelination secondary to the cortical neurolosis. Okay, compare with granulin and mapped. We saw that the symptomatic careers, the granulin mutation careers shows much more predominant superior longitudinal vasculitis and cingulum, and the mapped mutation careers shows more white matter degenerations of phonesis. And it's also connected with this symptom onset, the proximity to a symptom onset. As we mentioned, then the C9 mutation careers, there are more widespread cortical atrophy. There are also widespread white matter degenerations observed even in the asymptomatic C9 repeat expansion careers. See, then there's a lot, even in the asymptomatic stage. So the DTI shows a relative mutation-specific loss of white matter microstructure integrated in the asymptomatic stage of mini FTLD. Let's move to the fMRI. fMRI is used in the asymptomatic versus in the non-careers, in the mapped mutation careers. So you reported the first one is what we are in neurology about more than 10 years before. We see the decreased functional connectivity in the default mode networks. But a very recent studies has demonstrated that the coupling between the functional connectivity and cognitions was stronger for the mapped mutation careers compared to the controls. And this relationship increased when they're close to the expected symptom onset age. As the granular mutation careers also show the resting state connectivities, but we saw that also the asymmetric patterns in the parietal lobes, even if you the asymptomatic one. So for the C9, there are more recent state fMRI studies. It shows the salient network, the reduced functional net connectivity. Instead of the mapped mutation careers, we saw the default network. So here the somatomotor networks and dorsal attention networks were with age even the asymptomatic careers increased with the age in the asymptomatic careers, but reduced with age in the symptomatic careers. That means maybe some composite to in the asymptomatic of the functionals. So we move to the MRIs. So this is what we did. We applied the frontal lobes. We applied a voxel in the frontal, bilateral frontal MRIs to the mapped mutation careers and follow them now. And then this is a cross-sectional. We compare with the asymptomatic and symptomatic with the non-careers. We saw then there are some, even if in the asymptomatic map stage, we saw the markers NAA, which means indicates the neural loss. This is the neuronal markers has decreased in the asymptomatic stage. And this is what we did for the longitudinal MRI studies. In this study, we have the 6 mapped mutation careers from the posterior cingulate voxels, and we follow them longitudinally, follow them annually with a median of 77.75 years. And 4 of them, they converted. The 8 of them were converted from the asymptomatic stage to the symptomatic stage. So we saw then there is a point where the decrease in the NAA plus MI ratios and increased to about accelerated around 2 years prior to the symptomatic onset. So then we mentioned a little about molecular imaging. That this is what we did in the group did in the published in the 2015 in the Lancet Neurology. That means this, the post-mortem patterns of tau distributions, and we also can, this is a P301L mapped mutation careers, can be shown in vivo with this AV1451 tau PET. But this is a study from the Mayo Clinic by Dr. David Jones, which shows that this tau PET, different tau PET in the different kind of mutations, subtypes of the mutations. They use the AV1451s to show that whether these tau depositions can be shown in these different kind of submutations. You see that here within involved exon 10, we saw that here maybe a lot of them cannot show in the tau deposition there, but outside this exon without exon 10, we see that here the more predominant with this tau deposition. That means that what we used 1451, this tracers is more specific to the 3R, mixed 3R and 4R tau. For those pure 4R, which is maybe not so specific and off target, we can see here. And this is also see, this is pure 4R mapped. No, we cannot see any tau be shown by the AV 1451. The mixed 3R and 4R tau we see here, and this is more likely to be what we saw in the Alzheimer's disease. The tau is 3R and 4R taus here. But for more recently, we have two newer tau tracers is MK6240 and PBB3. They are thought to be more specific with wild ranging tau pathology and decrease the off-target bindings here. This maybe in the future we can use these tracers in our familiar FTLD patients studying. And FDG PET was shown in only the hypometabolisms in the asymptomatic mutation careers. We can see the different patterns here. And other molecular imaging technologies we know that there are DET scans, and when there are a lot of other tracers, we can show the symptomatic noise, neural inflammations, as well as the brain perfusions there. Where also a little of them, not a lot, applied in the familial FTLD. So, but in summary, we know that imaging biomarkers are vulnerable for accurate prediction of symptom onset and tracking the disease progression during the asymptomatic stage of familial FTLD. So identifying the molecular imaging biomarkers is important for the in vivo assessment of underlying pathophysiology changes and future disease-modifying therapies. But we know that the heterogeneous findings of familial FTLD studies might be expanded by the quite limited sample size, and the longitudinal studies with larger multiple cohort are needed in the future. And we now know there is a frontal temporal initiative that including the Europe and United States and everywhere that other countries. So that's what we need to do in the future. Thank you for your attention.
Thanks a lot for your excellent presentation. Okay, we'll have a discussion at the end. Thanks, Dr. Chen.
Thanks.
Okay, now it's my great honor to introduce our next speaker, Professor Jun Wang. Professor Wang is Vice Dean of the Faculty of Psychology and Deputy Director of IDG McGovern Institute for Brain Research at the National Key Laboratory of Cognitive Neuroscience and Learning, Beijing Normal University. She received her PhD in bioengineering from the University of Illinois at Chicago. Her research mainly focuses on cognitive rehabilitation after brain disorders, especially the structural and functional networks of the human brain using multi-modality imaging techniques in persons with Alzheimer's disease, mild cognitive impairment, and aging. The title of her talk is Beijing Aging Brain Rejuvenation Initiative, Revealing Neuroimaging Indicators. Welcome, Professor Wang.
Okay. Can you see my screen?
Yeah.
Okay. Good evening and good afternoon. So thanks for the kind introduction from Professor Zhang, and thanks for this unique opportunity for sharing our study with the international audience. So I'm Jun from the State Key Laboratory of Cognitive Neuroscience and Learning at Beijing Normal University, and my talk today is Beijing Aging Brain Rejuvenation Initiatives, BABRI, revealing neuroimaging indicators. So I will talk two parts. One is the brief introduction of our BABRI study, and the other one is the revealing neuroimaging indicators. So actually, the whole world facing the big challenge for aging. So the world population aging in 2017 reported globally, the elderly people aged over 60 will reach to 1.4 billion in 2030 and will be reached to 2 billion in 2050. But while in China, the elderly people aged more than 60 years old was 264 million in 2020, 2 years ago, representing 18.7% for the total population, and will be reached to 487 million in 2050. And the percentage you can see here is pretty high. It's around 30% of the total population. So therefore, many countries, they initiated the cohort studies of aging and cognitive impairment from different times, including the ADNI in US, and also Biobank in UK, and ELBO in Australia, and also Finger in Finland and other Asian countries like Japan and China. So in 2008, we initiate our BABRI study representing Beijing Aging Brain Rejuvenation initiatives. And our BABRI is a community-based longitudinal cohort study and focusing on symptomatic stages of dementia, aims to develop community-based prevention strategies for cognitive impairments and provides a platform for scientific research and clinical trials. And so far, our BABRI database in 2020, we collect over 10,000 participants aged over 50 years old and 60% for female subjects. And among them, 2,699 have been followed up at least once at 2 or 3 year interval. And we collect data from diverse categories, including 28 dementias, like medical history and cognitive function, and also lifestyle and habits, emotional status, genotyping data, and also biochemical markers, neuroimaging data, including MR scanning, and also PET imaging. And in terms of our BABRI design, and we have accelerated the longitudinal design of BABRI. So for the first 3 waves, we recruited the data from Beijing area. And since 2017, for the wave 4 and wave 5, we expanded to the other cities in China. So we collaborated with hospitals and also institutions in 10 provinces. So initially, we only collect data from Beijing communities. And in 2017, we expanded to the other cities around the whole China, and including Inner Mongolia, Shandong province, and also Hebei provinces, and also Guangdong province, and also the Qinghai province. And we called BABRI National Consortium Project right now, okay? And we published 2 review papers. One is in 2008 in China, in Science China for life sciences. And the other review paper we published in Alzheimer's and Dementia last year. And we introduce and highlight our research findings and also our applications and for the past years. And next, I will give you some interesting findings for our BABRI study, revealing the neuroimaging indicators. And first is the cognitive, we identified the cognitive aging trajectory for elderly people in China. And in 2014, we collect over 1,200 subjects from Beijing communities. And we identified the trajectory and the key points. Like at the age of 58 years old, we found episodic memory function has significant decline. And in the early age, at the age of 55 years old, the executive function has significant decline. And also we studied the other cognitive function as well. And in 2020, we published a paper in cerebral cortex, and we found we're using the DTI imaging study and also the graph-based analysis, and we found the integration of the brain structural connection, and they had significant decline in the frontal area and also parietal area and the superior temporal lobe. And this kind of network efficiency, they has the positively correlated with the cognitive function, including attention and also executive function. Okay. And the other one is to study the MCI-specific neuroimaging indicators. And we're using the functional MRI, and we studied the DMN network, and we found deactivation in the whole DMN networks, especially the anterior and to the posterior part. The functional connectivity decreased significantly, especially in the right hemisphere during the episodic activation task. And the other study, we studied how the age interaction with MCI. We all know age is the risk factor for the AD. And we found the gray matter atrophy, they have significant atrophy in the left thalamus area and the post-cingulate gyrus, and also in the right inferior frontal gyrus, and also right caudate area, and the central occipital cortex. And they all have the significant decrease with the gray matter volume. Okay. And the other one, we also studied the mechanism for the memory impairments in different subtypes of MCI, including AMCI and non-AMCI. And we found the hippocampal structural networks. And in MCI patients, they have severe and also the extensive damage in 3 networks. And also this kind of damage is related to the decline of the verbal memory and spatial memory. And we all know the anterior part for the hippocampus and represent for the verbal memory, and the posterior part for the spatial memory. Okay. And we also studied the subject memory impairment, MCI, SMI. SMI is also they tremendously increase the risk of AD, and we're using resting state functional MRI, and we found the connectivity between the anterior part to the posterior part. They decreased significantly for the SMI patients, and compared to normal control and AMCI, you can see they are in the middle of this process. And also for the different stage of AMCI, including mild AMCI, moderate, and severe, and they have different functional connectivity pattern. Okay. And also we revealing the brain mechanism of the risk factors, including diabetes, hypertension, cerebral infection, and in diabetes patients, and we found abnormal functional connectivity and in the superior frontal gyrus, and also for the median frontal gyrus. And also we found the abnormal white matter fiber damage, and also the functional connectivity reduction. Okay. And for the hypertensive patients, and we found the frontal parietal white matter networks, and they have the impairment. This kind of impairments, they related to the brain activation pattern and also mediated with the executive function. Okay, and for the cerebral infection, and we found that they damage the white matter, especially for the anterior part of the cerebral region, and it is related to the subcortical networks for function and structure. Okay. And the other part, we started investigate the genetic factor, how they related to the normal aging and AMC patients. And we found the ApoE carrier, and they have a severe atrophy in the parahippocampus gyrus, and especially for the TT carriers, they have significant decrease for the thickness and for the gray matter thickness. Okay. And also we studied the different genotypes like SPIN1, PCON, and SORL1, and all they have different modes for the functional connectivity, especially for the superior parietal gyrus and also inferior frontal gyrus for the functional connectivity. Okay. And the other very interesting study we published last year, and we studied the gender differences and the sex differences for how they related to the brain structure during aging. And we found in the whole region, they have this kind of atrophy. But for the female subjects, they have a more larger atrophy area compared to the male subjects. And also they have very they have a greater slope for the atrophy volume decrease. And also we studied the correlation between those correlated with the cognitive function. And we found the female subjects and also the male subjects, they have this kind of strong correlation with the memory function and also with the attention. And just last year, we published a paper in molecular neurodegeneration, and we provide the first literature review on the neuropsychiatric symptoms in AD, NPS. And we studied on-site times for the NPS. We found different NPS, they have different stage, occurs different stage of AD, such as apathy, depression, sleep disturbance, and also abnormal motor behavioral delusion, and eating disturbance, disinhibition, hallucination, and they all have different stage. But most NPS, they appear the symptom in the early stage, especially in preclinical AD or MCI, and they progress very severe during the whole progression to the AD. And also we studied a specific lesion pattern, and we found anterior cingulate area is the common damage area. And also the other part, like prefrontal cortex, especially the orbital frontal cortex, they are very region specific. They all related to the most NPS symptoms. And also we studied the PET imaging data, and we found the tau is the key driver for the NPS symptoms of AD. And compared to A beta and also the FDG, and we found that the tau pathology is superior, and they have the region-specific area, especially in the inferior temporal lobe. And also they can identify the NPS symptoms, including depression, apathy, and also aberrant motor behavioral appetite problems. And the other interesting study, we investigated how the education influence to the brain, and we found the elderly with higher education, they have better multiple cognitive function, such as memory function, language ability, and executive function. And also we found the anterior cingulate and also occipital forceps, they atrophy more slowly, okay, compared to the other part. And the last study I'd like to introduce is our traditional Chinese medicine. And this is a 2-year follow-up intervention. And we found that the TCM drug efficiency, and they changed the whole brain networks and enhance the precuneus connectivity. And you can see this is the 12 months data, and this is the 2 years 24 months data. They increased the connectivity significantly. And also for the other part, they also have this kind of change for the connectivity analysis. So over the past 14 years, we published over 120 peer-reviewed journal papers in diabetes, human brain mapping, in Journal of Neuroscience Radiology, all related to our neuroimaging study. And we got continuous funding support from our local government, and also from our National Science Foundation, Beijing local government, studying the neuroimaging indicators. And for the international collaborations, we signed agreement with Beijing Alzheimer's Institute in 2008. And also in 2018, we collaborated with UC Irvine to establish a joint center for brain aging. And over the past 14 years, we published more than 50 journal papers together. And recently, we collaborated with King's College London, and our collaborator, Professor Asland from Institute of Psychiatry, Psychology, and Neuroscience. And we focus on the gender differences in dementia from cross-cultural perspectives. And in the middle, this figure is our founder of our BABRI study, Professor Zhang, and he is also our director of the BABRI Center. And the other part I'd like to introduce is our application, because besides our basic research, we translate our basic research findings into applications. So we launched our cognitive assessment system and also brain health screening and cognitive training online platform. So you can use a computer, an iPad, and also mobile phone to get this brain health screening. And also we can use those electronic system, and we can have cognitive training online platform and to do the training games and to help the people, help the elderly to improve the memory function, language function, executive function in 7 cognitive domains. So for the brain health screening programs, and we have 3 tiers, 3 different level programs. And one is we called BABRI screening programs for memory clinics. So this is a very fast and rapid assessment. It's around 6 minutes, and including mini MSI, EMT, and subjective evaluation. And the second level we called BABRI dementia risk screening programs took around half hours, and to investigate, assess the executive function, episodic memory, language ability, etc. And if you have the high-risk population for MCI, so we collaborated with hospital. So we sent our elderly people to the hospital to collect imaging data and also blood and gene detection and for the imaging programs. And because our successful online platform, so the Beijing Municipal Health Commission support us to perform Beijing Brain Health Examination Program. We also called dementia risk screening for the elderly people in Beijing area. So in Beijing, also we have the report at the end of last year. So in the whole Beijing area, so the elderly people aged more than 60 years old, right now is the number is 4.4 million, and the percentage is the first time to reach to 20%. So it's the highest percentage of the total population in Beijing area. So we face a very big and severe challenge. So the first 2 years we collect the data only in the 6 districts in the Beijing area, but last year we expanded to the whole 16 districts in the whole Beijing areas. So from 2019 to 2021, so we already collect 487,000 elderlies from the Beijing communities, involving 1,523 communities. And thanks for their support. And accept applications and research, basic research, and also we promoting brain health screening program nationwide. And we have WeChat platform, and we provide this kind of knowledge to let the audience to know what's the brain science, what's the neuroimaging study, and what's the Alzheimer's disease. And also we organize BABRI online seminars every week. In Thursday, just right now, so we at the same time, and we have 162 seminars online, and we invite different scholars from different hospitals and from different institutions to share the knowledge and to share the ideas and research, advanced research findings with the audience, and to let the people know our BABRI study. So if you are interested in our BABRI study, and if you want to join our BABRI study, and you can contact with me directly by this email. So thank you very much, and thank you for listening.
Many thanks for your presentation, Professor Wang. Okay, our next presenter is Mengya Xing. Dr. Xing is an associate professor of neurology in Tianjin Medical University. She's a member of the Psychiatry and the Mental Health of the Chinese Geriatrics Association and a member of the Brain Cognition and Health of the Chinese Society of Gerontology and Geriatrics. The title of her talk is Cerebral Blood Flow Pattern Mirrored with Arterial Spin Labeling in Subcortical Ischemic Vascular Dementia.
Okay. Thank you, Professor Zhang, and good evening, everybody here and colleagues overseas. It's my honor to have this opportunity to address the speech about what we did with arterial spin labeling in dementia. We know that changes of cerebral perfusion play an important role in the development of dementia. Perfusion is the passage of fluid through the circulatory system or lymphatic system to an organ or tissue, usually referring to the delivery of blood to a capillary bed in tissue. And CBF, with the abbreviation of cerebral blood flow, is the blood supply to the brain in a given period of time. There are many methods to obtain the perfusion pattern. For example, we can use CT scan, inhaling xenon 133 scan, and also we can use SPECT with 99 MTC labeled radiopharmaceuticals and 015 water PET, which is the gold standard for CBF assessment. And we also have methods of MRI, such as dynamic contrast enhanced MRI and ASL MRI. ASL offers perspective of a non-invasive assessment, CBF assessment of brain perfusion, which making it potentially very useful in research and clinical studies. And furthermore, ASL is very useful for tracking CBF changes over time due to recovery and therapy. So how can we obtain ASL? ASL uses artery blood water as an endogenous tracer. We can tag inflowing artery blood by magnetic inversion. We acquire the tag image. We can see the image here. The images are not very clear. And then we repeat the experiment without tag. We acquire the control image. We can see the images here are not clearly enough to see what happened. Then we got the subtract of control image and the tag image. The difference in magnetization between control and tag conditions is proportional to regional cerebral blood flow. Now we can see what ASL images is. So we obtained ASL. How can we analyze them? Both univariate analysis and multivariate analysis can detect what happened in brain function. But univariate analysis shows regionally specific changes, always ignores the functional correlations between anatomical structure of interest in the brain function. It was early recognized that on those neurodegenerative processes, even it's highly localized, are associated with disease-related, disease-specific alterations in functional connectivity across the whole brain. So we use scaled variance pattern in the brain function. This picture we can see is the computational steps of SSM-PCA. There are many types of dementia. And each type of dementia have its own neuropathologic characters. For example, the main neuropathology of AD include A-beta and tau, which we can also observe in Lewy body related dementia and FTD respectively. And also there are several overlapping pathogenesis between AD and BAD, such as endothelial dysfunction, BBB breakdown, and hypoperfusion. Increasing evidence indicates that with ASL, we can identify AD from other types of dementia. And also, furthermore, AD can explore the overlapping pathogenetic mechanism in dementia. So we discuss several situations. First, we evaluate the age-related changes. We can see that after normalization for global value, we can see that regions with decreased CBF in aging is bilateral superior frontal gyrus, right middle frontal gyrus, left superior frontal gyrus, bilateral inferior frontal lobule, and right caudate body, left superior temporal gyrus, and bilateral cerebellum. Regions with relatively increased area, including bilateral middle cingulate gyrus, left middle orbit frontal gyrus, bilateral putamen, and bilateral fusiform gyrus. To identify the age-related CBF pattern, we applied SSM PCA analysis. As we can see, the regions in relatively decreased in frontal and parietal areas and the cerebellum, and relatively increased areas include temporal and occipital areas. Then we explore the VBM changes in AD patients. We can see that before ANCOA, in AD patients, there are absolute decrease CBF in bilateral middle occipital gyrus, bilateral inferior parietal lobule, bilateral inferior temporal gyrus, and bilateral posterior cingulate and precuneus. After ANCOA normalization for global value, we can see that relatively decreased CBF in bilateral inferior parietal lobule and bilateral inferior temporal gyrus, bilateral middle cingulate and posterior cingulate cortex and precuneus. Relatively increased regions include bilateral precentral and post-central gyrus, right supplementary motor area, and bilateral putamen. To identify the AD-related pattern, we also applied SSM PCA. We can see that the covariance pattern shows that the relatively decreased area include middle and posterior cingulate and precuneus, inferior parietal lobule, modal frontal gyrus. And the relatively increased areas include putamen and a few frontal areas. This covariance pattern is high consistent with FDG PET result of typical AD, which makes ASL of AD makes an alternative of FDG PET. We also enrolled another independent cohort for identification of ADRP for validation of ADRP expression. We can see that subject expression score in AD are higher than HCs in either identification cohort and validation cohort. And the ROC curve was generated for subject expression score and to determine the optimal cutoff value for distinguish AD from HCs. We can see that the AUC in identification cohort and validation cohort are nearly the same. And identification cohort have a moderate sensitivity and high specificity. In validation cohort, the sensitivity and specificity are really high. Which means our covariance pattern is stable and very can make an optimal cutoff value to distinguishing AD from HCs. We also explored what happened in AD about cognition function with subject expression score. We can see that there are predominantly decrease in HC than in AD than HCs. And in identification cohort, global cognition and all cognition domains are strongly negatively with subject expression score. But in validation cohort, all cognition domains except memory and language are moderate related to the subject expression score. We've just mentioned that there are some overlapping pathogenesis between AD and SFAD. So we explored what happened in SFAD. After proportional normalization for global value, we can see that regions relatively decrease in SFAD include right angular gyrus, right superior parietal gyrus, bilateral inferior frontal gyrus, right superior occipital gyrus, bilateral middle occipital gyrus, and superior cerebellum. Regions with relatively increase in SFAD include right superior frontal gyrus, right precentral gyrus, right Rowlandic operculum, and bilateral putamen, bilateral thalamus, and bilateral pallidum. To identify the SFAD pattern, we also applied SSM PCA analysis. We can see the covariance pattern showed here. The relatively decreased regions include frontal, temporal, and parietal areas. We also enrolled another independent cohort to identification for VDRP and for validation of VDRP expression. We can see that the ROC curve in identification cohort and validation cohort, the cutoff in the two cohorts are almost the same, and either identification cohort and validation cohort has high sensitivity and specificity, which makes this pattern a stable and optimal cutoff value to distinguishing SFAD from HCs. Now we have discussed the ASL covariance pattern of ASL with aging, AD, and SFAD. We found that ASL is very useful in diagnosis in neurological and psychiatric diseases. Multi-center and longitudinal studies are worth conducting with optimized and standardized ASL to elucidate the diagnosis and facilitate the early diagnosis of neurodegenerative diseases. That's all. That's what we did with ASL in dementia. Thank you for your listening.
Thank you so much. Now is our Q&A session. Does anyone have any questions for our speakers? Okay, Dr. Xu.
Good evening, Professor Wang Jun. I'm Dr. Xu from Tianjin Medical University General Hospital. I have one question about APOE4. It's well known that APOE4 is one of AD risk factors. In APOE4 carriers are more susceptible of AD and usually endure more severe cognitive impairment than non-carriers. However, some researchers reported that among MCI or AD patients, the cerebral blood flow of some increase in some brain regions of APOE4 carriers than non-carriers. Is this phenomenon may associated with the enhanced neuronal activity. So my question is, how do you think about the role of ApoE4 in AD spectrum? Whether it plays opposite role in different stage of AD? Thank you.
Thanks for the excellent question. Actually, ApoE, they have the mixed findings. So some findings, they have this kind of, a strong correlation ApoE with the median temporal lobe atrophy, especially the hippocampus atrophy and anthonic cortex. So, but the other studies, they found they had no sort of this kind of correlation with the medial temporal lobe atrophy. So the findings is quite mixed. So I can't tell you, how the ApoE carrier. So we can say ApoE carrier, they increase the risk of the AD onset, but we cannot identify, this kind of, you mentioned, the opposite AD roles in the whole AD spectrum. Yeah, because in our study, so we found the normal cognitive elderly with the ApoE carrier, so they have the very significant atrophy in the parallel hippocampus gyrus. And this is in the early stage, but the whole spectrums, because we haven't studied the whole longitudinal data yet, so I can't tell you the whole spectrum how they change. Thank you.
Thank you, Professor Wang. Other questions? Dr. Xing.
Thank you, Professor Zhang. And I have a question to Professor Chen. Thank you, Professor Chen. Thanks so much for your excellent report today with lots of key points about the neuroimaging in FTLD. And now my question is, as you mentioned, ischemic cortical atrophy was observed in FTLD patients with granulin mutation, which was found early, not obvious in MAPT or C9 mutation. So how to explain this phenomenon and what's the possible significance about this? Thank you.
Thanks. This is a very good question. We know that, as I mentioned before, then the asymmetric atrophy is a very common findings in the granulin mutation carriers. They are asymmetric in both left and right asymmetry was observed, even if in the same family. So, but the underlying causes for the asymmetry remain still unknown. But compared with MAPT and granulin, we know that granulin is loss of function. They are related with TDP43. They were related with tau pathology as well. But MAPT is only with the tau pathology, and we know that the C9 always with this TDP43 pathology, right? So the underlying protein is different. But as we observed during current studies, we know that granulin has asymmetric early in the asymptomatic stages. But I remember then there is studies for the longitudinal studies by Dr. Whitwell in 2015. I think her findings that the symmetric rates of entropy through longitudinal, we measure rates of entropy, right? So the rates of entropy were symmetric in the FTLD patients with granulin mutations. So that means that the possible explanations for the discrepancy is that the asymmetry in the rates of entropy may be more apparent early in the disease stage, but becoming more and more symmetric over the time in the disease durations, as we were noted previously. So actually only then the specific phenotype and associated with the lobar cortical atrophy changes maybe related with different sensitivity and specificity of analysis methods as well. As mentioned, we used the TBM thing, right? So we use the lobar volume combined as a composite together. But some studies using the voxel-based morphometries, so they didn't get any positive results. Maybe just because this asymmetry, then you dilute any of these abnormalities by left or right predominant. By combine them together in the group levels, so you cannot get any results from them. So that's what my answers. Hope that you are satisfied with this answer. Thank you.
Thank you. Thank you. I gained a lot from your answers. Thanks again. And next question is Wang Jun. Thanks,
Okay.
Wang. Thanks, Professor Wang, and thank you so much for your wonderful report about the neuroimaging indicators. And we've gained a lot. And my question is about the white matter hyperintensity, that is to see WMH. Studies have found that the plasma A-beta concentrations were associated with WMH load in AD and MCI patients who carried ApoE epsilon 4 allele. So what's your view about the role of WMH in AD? Thank you.
Thank you for the great question. So actually, in recent years, WMH has become a very powerful factor related to cognitive decline. And the research shows that WMH distribute in the anterior part of the brain is related to the decline of the executive function during aging. And while the WMH distribute in the posterior part of the brain is more common in AD. So although the observational and pathological study have shown that WMH may originate from ischemia and can be maybe caused by the continuous and insufficient perfusion. So there is new evidence that they may also reflect the deposition of A-beta in the blood vessels, especially when they're distributed in the posterior part of the brain and exist in AD patients. So that's my answer. Thank you.
Thank you. Thank you again.
Mm-hmm.
I have a question for Professor Xing. Professor Xing, thank you for your excellent lecture today. We know that in previous studies, we know that the production of SIVD image markers seems to be different. So whether hyperperfusion plays a similar role in the production of different markers.
Thank you for your question. I think it's a very good question. I think the decreased BF is the major cerebral hemodynamic alternation in SIVD and pathologies, which cause reduction in global CBF, such as atherosclerosis and atherostenosis are involved. Most of SIVD is caused by cerebral hypoperfusion, and after cerebral perfusion, the subsequent endothelial dysfunction could lead to many processes, such as increase in BBB permeability and exposure of neural cells to harmful substances, a rise of inflammatory environment, and dysfunction of neurovascular coupling. And the result is activation of glial cells in the CNS, which lead to WMLs and neural damage. I think the biomarker can divide it into some groups, such as biomarkers of inflammatory response, biomarkers of cerebral neuronal neural system tissue injuries and biomarker of coagulate and thrombosis and circulative mRNAs, and also some image biomarkers. And these biomarkers may be observed in different times during the sequence after hypoperfusion. I think the hypoperfusion as an initiating factor for SAVD, but the interact with biomarkers will be further and confirmed in future studies. Thank you for your question.
Okay, thank you. Any other questions? Okay, Dr. Lee.
Thank you, professors. Thank you, professors, for your presentations. It's my great honor to learn from you. And I have a question about the cognitive screening for Professor Wang. As we know, there are many neuroimaging techniques for cognitive evaluation. And the first question is, which neuroimaging techniques are more sensitive to screening for early cognitive decline? And the second question is, which neuroimaging biomarkers can be used to predict the progression from MCI to AD in clinical research? Thank you.
Thank you for the tough question. Thank you. So the first one is for the executive function decline. I think, yeah, because over the past 14 years, and we have different assessments. And I think for the executive function, so this is very sensitive, yeah, for the cognitive function assessment, yeah, for this early detection, yeah, for the early stage of AD. And second question is, what's the earliest? Sorry for the second question. Yeah.
Wait. Oh. And the second question is, which neuroimaging biomarkers can be used to predict the progression from MCI to AD in clinical research? Thank you.
Okay, from my point of view, so I think the functional networking analysis is more, sensitive, yeah, compared to the structural connectivity analysis. Yeah, so I think the A-beta labeled PET imaging, so the analysis of SUVR so might be very sensitive, yeah, for prediction of MCI progress, yeah, to AD. So this is my point. Yeah. Mm-hmm. Thank you.
Thank you for your explanation. Thank you.
Mm-hmm.
Okay, thank you. I think I have a final question for Professor Chen. You mentioned about some novel tau PET tracers. So do you think it's also sensitive? I mean, the second generation tau tracers also sensitive to other tauopathies beside Alzheimer's disease?
Oh, thank you. Thank you, Professor Zhang. This is very important questions. This means it's guiding us what we used for our current studies in the FTLD patients. Yes, I think I didn't describe it in my slides in detail for the two recent studies there. So for the I think the second generations of the tau tracers, it seems to be more sensitive and more specific to the tau. And so in the current studies, they have shown that even if there's the tau mutations, which is subtype with P301L, which cannot be shown by the AV1451, but it can be revealed by the tau deposit even early in this asymptomatic stage by the MK6240, as well as the PBB3. They have their study reported there are two groups there. One is within 1 year, the short disease duration, and more than 1 year, the longer non-disease durations. These two groups, they also showing that with this second generation tracers PBB3, then tracers, you can see then the tau depositions even in the 3R and 4R. It's not the Alzheimer's disease tau. It's other types of tau. You can see then it's already deposit there and increasing with the age and with this disease durations. So I think these two might be more, in my opinion, it's more specific and more useful for our FTLD patient studies. Thank you.
Okay, thank you. Thank you so much. Okay, I think that's the end of our workshop today. I greatly appreciate our speakers' wonderful presentations and the attention of all of the members of the audience, both those who are here in person and those who are participating remotely. Thank you all. Good night and have a good day.
Bye. Thank you.
Thank you. Bye-bye. Have a good
Bye.
night.
Yeah,
Good night.
Good night.
Thank you.